Hello, thank you for coming to see my poster. Today, I'm going to show you the results of a drug screen that has identified novel compounds that stimulate type 2 collagen in human chondrocytes. So why do we care about type 2 collagen? Well, type 2 collagen is the primary component of articular cartilage and is produced by chondrocytes. Cartilage degeneration is the hallmark of osteoarthritis, a debilitating disease that affects millions worldwide. Our hypothesis is that stimulating type 2 collagen production in chondrocytes can reverse or prevent osteoarthritis, and that this can be a novel therapy for the disease. So in this study, we have used a novel 3D chondrogenic assay to successfully screen 390 natural compound library and have identified novel compounds that stimulate type 2 collagen. So how does our novel assay work? Well, it's based on a type 2 collagen promoter and Gaucher luciferase construct that's transduced in primary human chondrocytes. So when type 2 collagen is stimulated in the cells, Gaucher luciferase is produced and secreted into the media. We can then sample the media to quantitate the amount that's secreted and relate it back to the amount of type 2 collagen produced. We plated our transduced cells in a 96-well non-adherent plate to induce pellet formation. We treated the pellets with compounds from the natural library and assessed chondrogenesis for 22 days. During this time, we regularly imaged the pellets, sampled, and replaced the media. Results from the screen can be seen here in Figure 1, with each row representing one of the compounds and the blocks representing days where luminescence was assessed. Blue blocks indicate readings that were at least twice that of the control, while the red represents luminescence that was 1/1000th that of the control. Candidates that were selected for further study are highlighted by the black boxes. Graphs of raw luminescence as well as normalized luminescence can be seen here in figures 2A and 2B for our top candidates. We also stained our top candidates for type 2 collagen expression as well as glycosaminoglycan content as seen here in figure 2C. As you can see, there's a substantial increase in type 2 collagen production in spheroids treated with compound 84, with no negative impact on glycosaminoglycan production. To validate the compounds selected from the screen, we ran our assay with multiple replicates, and the results are shown here for 23 days of the experiment. The data confirms that compounds 84, 204, and 418 have increased luminescence as compared to the controls, although only 204 and 418 showed any statistical significance. We also tested multiple concentrations of compounds 204 and 84, and from that data derived drug response curves that suggest an optimal concentration less than 10 micromolar for both compounds. We do not have data for compound 418 as we have not been able to source it. Altogether, our data supports that our novel 3D assay can be used as a tool for high-throughput screening and is an improvement over traditional methods as it is non-destructive, easy, and allows for temporal monitoring of type 2 collagen. Furthermore, we have identified and validated 3 new potential compounds for stimulation of chondrogenesis, and future studies will identify drug targets as well as assess their drug potential in vivo. With that, I would like to thank the members of the Keene Lab as well as my mentor, Dr. Keene, for all their help in this project.